right. Looks like we have a pretty good crowd now, so I'm gonna super excited to have Andy McGuire here. He did his PhD at University of Guelph in Ontario. Is that pronounced right?
Yep, you nailed it.
And then moved here to Seattle for a postdoc in Leo Stamato's lab at Seattle Biomed, and then eventually the Hutch, where you then graduated into an assistant prof position, focused kind of along the way on, a bunch of different really interesting stories related to vaccine design, antibody discovery, and kind of novel methods to, elicit antibody responses directed at, specific viral antigens. A lot of the work is focused on initially on EBV and HIV infection, but now you kind of branched into the more recent and exciting papers into SARS and RSV, I guess. So excited to hear more about your work, Andy, and I think the center is as well. So thanks a lot for joining us and spending the morning talking to us, and excited to hear this seminar.
Yeah, thank you, Rich. Thanks for the introduction. It's been great to meet some of you, and I'm looking forward to meeting some more of you afterwards. Rich mentioned we did do a fair bit of SARS-CoV-2 work over the past couple of years, but I'm not going to speak about that today because I think we're probably all a little bit sick of hearing about it. So I'm going to talk about two different things in my talk today. The first thing is going to be our work to develop anti-idiotic antibodies as novel vaccine antigens to elicit broadly neutralizing antibodies against HIV, and in particular trying to target germline antibodies. And then for the second part of my talk, I'm going to talk about another research area, and that's where we're working towards defining and eliciting protective immunity against EBV. So to begin with, for the HIV portion of the talk, it's important to understand the HIV envelope protein. Of course, this is it's the sole virally encoded protein on the surface of the HIV-1 virion. And it serves to both mediate attachment to the host cell through CD4, and it also functions to promote the merger of the host and viral membrane during entry. So it plays a critical role in both aspects. One of the problems that's sort of plagued HIV vaccine development is that the envelope itself is extremely variable in sequence. That's probably one of the most diverse viral glycoproteins out there. But despite this diversity, there are broadly neutralizing antibodies that develop in a subset of people who are HIV infected that can bind to multiple different HIV envelope strains despite this diversity and block HIV-1 infection. Importantly, passive transfer of broadly neutralizing antibodies can protect against experimental HIV infection. And to some degree, even in humans, they've now done passive delivery of neutralizing antibodies as a preventative measure in a phase 1 trial. So we think if we can elicit these types of antibodies at high titers in humans, they should be able to protect against HIV-1 infection. But so far, efforts to elicit them through vaccination have not been successful. And interestingly, people have cloned out monoclonal antibodies from HIV-infected people and identified actual monoclonal broadly neutralizing antibody specificities on the spike. And so shown here are sort of a map of all of them. But what's interesting about them is the spike shown here in gray. The antibodies that are broadly neutralizing tend to target these sort of conserved sites of vulnerability. There's one at the apex of the trimer. There's one that's focused on the V3 glycan patch on near the top. Antibodies that target the CD4 binding site are seem to be highly prevalent. And then there's antibodies that bind at the interface of the two components of the envelope, the GP120, GP41. And these often encompass the fusion peptide. And then there's another site of vulnerability at the base of the molecule. So antibodies can, of course, bind anywhere, but these are the sites where the neutralizing antibodies tend to bind. And what's interesting is a lot of these BNAbs have shared genetic and, as a result, structural features that allow them to interact with the envelope in this way. And so the one thing to consider in the way that we think about how these develop in the context of infection is how they first come about and then how they develop. So if you'll recall, antibodies are produced through random antibody VD and J recombination to produce an antibody heavy chain, and that analogous process happens on the light chain, and those compare randomly to form an antibody that's initially expressed on the surface of a naive B cell and associates with some signaling molecules called the B cell receptor complex. And this allows the B cell to respond to antigen when it's bound by the antigen binding region at the top of each arm. But what's important to note here is that the sequence of the antibody is initially encoded by the chromosomal sequence of the different antibody genes. I don't know how to get rid of this little thing at the bottom. There it goes. So in the context of HIV infection in an individual who developed BNAbs, what would happen is one of these B cells with a chromosomally encoded, rearranged naive B cell receptor would recognize its cognate antigen, in this case, the Hiv envelope protein. That triggers the B cell to become activated, and it triggers it to start to divide, and it introduces mutations into the antibody genes through a stochastic measure through a pre-programmed enzymatic step. And the result of this is that you end up with a pool of daughter B cells that have acquired mutations in the B cell receptor repertoire, and this creates a diverse pool of cells that can then compete for antigen. So what happens next is the B cell that has acquired a gain of function, so this one up here, this mutation allows it to bind to the envelope better. And as a result of that, it can compete for T cell help and antigen in germinal centers, and it will go on to survive, whereas the other lower affinity B cells will be selected against and eventually die. In the context of HIV infection, which is chronic, and over the course of several years, this can happen over and over again, eventually leading to the acquisition of even more somatic mutations on the B cell receptor that results ultimately in higher and higher affinity antibodies for the antigen. And these will eventually become antibody-producing B cells and secrete the broadly neutralizing antibodies that we're hoping to elicit through vaccination. But as a result of this affinity maturation process, in particular in HIV, because it is such a chronic infection that goes on for so long, what happens is the antibodies accumulate a lot of these somatic mutations. And so what I'm showing here is at the bottom is a sequence of a broadly neutralizing antibody called 3BNC117. So this was isolated from an HIV infected donor from a monoclonal B cell. And we know the sequence of that antibody. And when you can take that sequence and align it to and blast it against the antibody chromosomal genes, we can say that this one came from VH12, in particular, it came from VH12 gene. And I've shown the sequence of the VH12 gene at the top and highlighted all the mutations that have accumulated over time in this developmental process. And just at the bottom, I'm showing the structure of both the germline antibody, what we call the germline antibody. So as it's encoded by the sequence at the top, compared to the mutated version of the antibody. So after it's acquired all its mutations, and the paratopes indicated by this dashed line. And you can see that there are some changes to the overall shape of the antibody, as well as changes to the electrostatic surface potential, which are indicated in shades of blue and red. So this change that happens as a result of the affinity maturation process actually has pretty significant consequences for the way we think about vaccine design. And I'm trying to illustrate that here on this slide. And so what this is showing is each dot represents a different broadly neutralizing antibody against HIV. And the bottom, across the bottom are different recombinant HIV envelope proteins. And so the farther up the y-axis the dot is the stronger it binds to that particular envelope protein. And consistent with the idea that these antibodies can bind and neutralize diverse viral strains, they can recognize most of these recombinant envelopes. But what's remarkable is if you take the sequence of these antibodies and revert them back to what we think is what the germline form, so we align them to the various VD and J genes that they're most closely aligned to, and then remove all the mutations that have been acquired through somatic mutation, they completely lose their ability to bind to the HIV envelope proteins. And so this is not only narrow phenomenon that we observe with this handful of antibodies that I'm showing you here, but it's been largely observed for most of the broadly neutralizing antibodies that have been discovered for a number of years now. And what we think this means is that in order to develop a mutated broadly neutralizing antibody, the process has to start with the engagement of a naive B-cell receptor in its germline configuration. And what these observations suggest is that this simply is not happening. So the run-of-the-mill recombinant HIV envelopes that we have available to us that might be tested for vaccines, although they can bind the mutated envelope, the germline is considerably different, and then it no longer is able to interact with this envelope. And so what this suggests is that we need immunogens that can specifically target these precursor B cells, and those could be HIV envelope proteins for the most part. And this has sort of led to the development of a fairly large field called germline targeting. And there's been sort of three major approaches that are in various stages of development to target these germline B cell receptors. One of them is called Lineage-Based Design. And I'll try to briefly explain this as best I can, but basically people who are newly HIV infected are enrolled in a prospective cohort. And then there's longitudinal blood samples taken from these people over several years. And then the hope is that somebody in that cohort will develop broadly neutralizing antibodies. And then you can isolate a monoclonal from that infected donor and isolate a monoclonal BNAb. And then once you have that sequence, you can go back and deep sequence the repertoire that's present in those longitudinal samples that were drawn, and hopefully track the development of that BNAb lineage over time. And go back and actually identify the point at which that broadly neutralizing antibody was first observed, and it will be unmutated in this case or in its germline configuration. And then what often happens in these cases is that they can sequence an envelope that's co-circulating at the time that the BNAb lineage emerged and identify an envelope clone that can engage it. And what's interesting about this is that oftentimes they will not be able to, I don't know what Thor is asking me here. Okay. But they won't react with other heterologous envelopes. I don't know where those lines came from, but I'll just move on. The other approach is structure-guided design, and this is where you can sometimes observe differences between the structure of a BNAb bound to envelope.
Andy, I think it's if you go to the left side,
Yeah,
like your mouse is doing it. So if you go to I think it should say something about ask to annotate, or yeah, leave it like that. That's just like the cursor, like the pointer. And if you have it in that configuration, you can use it as a pointer. And sorry about that.
That's okay. That's fine.
WebEx.
Yep, technical difficulties. So the other approach is, one of them is structure-guided design. And in this case, what happens is somebody can solve the structure of a BNAb bound to an HIV envelope protein. And then by looking at the structure and inferring what the germline sequence that antibody was, they may be able to identify, for example, a clash with the germline antibody that's not present in immature, and then somehow modify the envelope so that it can engage that germline BNAb. And then the last one that's widely used is Mutagenic Display. So in this case, you can create and screen mutant libraries of an HIV envelope protein, by either yeast or mammalian surface display, and then bait those with the germline antibody of interest to identify an envelope clone that can bind, and then produce that as a recombinant envelope. And so with all of these approaches, they're based primarily on the HIV envelope protein itself as a starting point. And so what we've been thinking about is looking at it in a different way, and that is developing anti-idiotypic antibodies as a way to target these germline B-cell receptors. And the idea is basically that if you have a naive B-cell with a B-cell receptor that you know you want to target, it has some genetic features that you want to target, you can make it as a soluble antibody, immunize mice or rabbits or some other animal, and they'll mount an immune response against it because it's a foreign antigen. And then we can make hybridomas from those B cells and then screen those for what we call an anti-idiotype that binds the paratope of the antibody we want to target. And then the idea is we can use that as an immunogen to stimulate the naive B cell as a way of activating it. And it gets around some of the other issues with the approaches I discussed earlier, and then it doesn't require structural knowledge to guide vaccine design, and it doesn't require on large longitudinal cohorts to do this kind of germline targeting approach. And so this was some work that we pioneered with Justin Taylor and Leo Stamatatos shortly after we came to the Hutch. But there's another potential advantage to this approach that is if you consider you have, in the context of a vaccine setting, you'd have a pool of naive B cells that could potentially respond to any given vaccine, and then those is your target B cell that you want it to activate. You could immunize with one of those germline targeting immunogens that's based on the envelope protein, and it will be able to engage the target B cell because you've designed it to do so. But it can also have these other off-target epitopes that other B cells are going to recognize. And so what we think will happen is you'll expand all of those B cell clones, and depending on the relative abundance of your target cell and the off-target B cells, as well as the abundance and relative affinities, they're going to compete in germinal center reactions, and you may end up outcompeting the cells that you actually wanted to target. In contrast, if you have that same target pool of B cells and you immunize with an anti-idiotype that was specific for your target B cell, we think you can engage the target B cells with pretty high affinity and specifically activate them. And of course, you're going to get some off-target responses to other portions of the anti-idiotype. But what we think will happen is that it'll have an advantage over an envelope regimen where if you imagine that you can prime this B cell, but you need to immunize probably several times to achieve the level of somatic mutation that we see in natural infection to develop BNAbs. So you'll have to immunize the second time with envelope. And what we think might happen is then you expand B cells further, and you may boost some of these off-target responses that could further outcompete the target cell of interest. In contrast, if we primed with the anti-idiotype, we've expanded the cells that we want to expand, and we think they may have a selective advantage if we then immunize with, say, a germline targeting immunogen that will look more native-like, but now those cells might have a competitive advantage because they had a head start because they were primed with the anti-idiotype. And so that's the idea behind the whole approach. And so far for HIV, we've been focusing on a particular class of antibodies called VRC01. So I mentioned earlier that a lot of the broadly neutralizing antibodies that target specific epitopes have genetic features, and I'm going to show you a very clear example of one here, which is the VRC01 class. And so these antibodies have been isolated from at least 12 different donors at this point. And this table sort of summarizes them. So the donor from which the BNAb was isolated is listed on the left. The name of the monoclonal itself is listed here. And then there's some information about the genetic makeup of those antibodies. And so you can see that all of them use the VH1202 antibody heavy chain gene. So they all have that in common. And the reason for that comes down to the structure of the VRC01 class antibodies. So what I'm showing here is an alignment of 9 different VRC01 class antibodies that have been isolated from 5 different donors, and they're all superimposed on each other. And they're bound to the GP120 protein of the HIV envelope protein up top. And you can see that they have nearly identical angles of approach and almost identical structures. There are some subtle differences because they have all undergone different developmental pathways, but overall they have the same sort of mode of interaction. And one of the keys of that interaction is this region called the CDRH2, and that's shown here in green. That makes important contacts with the GP120 CD4 binding loop on the envelope up top. And so that CDRH2 region is encoded by the VH1202 gene, and there's particular amino acid contacts that are made that are specifically found in the VH1202 gene. So that key piece of the interaction is encoded by VH1202. And the other key feature of VRC01 class antibodies is what's known as having a 5 amino acid CDRL3. And so this is the part of the antibody that's formed when the V and J junctions come together on the antibody light chain. And so normally that process results in CDRL3 of 9 amino acids long. And in VRC01 class antibodies, they're always 5. And so that CDRL3 on VRC01 class antibodies is shown in this cyan loop here. And so that it's basically a loop that protrudes out and makes part of the antigen binding face of the antibody between two beta strands. And you can imagine if this were a normal length CDRL3, so it was 9 amino acids longer, it would protrude up further into the GP120 structure and clash with elements of the loop D and V5. So it has to be a short 5 amino acid CDRL3 to be able to accommodate this mode of binding on GP120. And so these two key features, which would be present on a naive B cell in a chromosomally encoded germline configuration, means that we think if we can target these types of B cells with our anti-idiotypes, all of those B cells would have the potential to become VRC01 class antibodies further down the road. Don't know how to make this go away. So we set out to do this, and what we did is we immunized mice with either the inferred germline version of VRC01. So as I mentioned, all of these antibodies were isolated from infected individuals, but we aren't 100% sure what they actually looked like in their germline configuration. So what we can do is take the sequence and align it to the closest chromosomal genes and then remove all the somatic mutations. But we can't really do that in the CDR regions because they're harder to predict. So this is sort of our best guess as to what the germline of VRC01 looked like. And we immunized mice with those. And then we also immunized another group of mice with two other inferred germline versions of VRC01 class antibodies called 12A21 and 3BNC60. We then screen them for binding to a panel of inferred germline VRC01 precursors. So again, these are the reverted versions of true VRC01 class antibodies isolated from infected donors. We also screen against a panel of what we call real VRC01 precursors. And so these are obtained through high throughput unbiased sequencing of naive antibody repertoires. And we've done by other groups, primarily Brandon Dukoski and George Georgiou. And we were able to look through their data and find antibodies that had a VH12 heavy chain with a 5 amino acid CDRL3. And so we made those as recombinants and tested them for binding as well. And then we have a panel of control antibodies that include some of the mutated forms of the germline VRC01 antibodies, as well as a bunch of other inferred germline HIV neutralizing antibodies that are not VRC01 class. And so you can see that the red, the darker the shade of blue indicates the stronger the binding of any idiotype to a given antibody. So each idiotype is shown here on the left. And some of them, for example, IV1, 2, and 3 that we call them, really only have affinity for the inferred germline of VRC01. So these are the B cells, or the antigen that those mice were immunized with. Whereas some of the other ones show a little bit broader reactivity, so they can bind to more of the real and inferred germline precursors of VRC01 class antibodies, and some of them, and most of them show little to no reactivity with the control antibodies. The exception are these two antibodies, IV9 and IV12, which show binding to the inferred germline of an antibody called IOMA. And that's interesting because IOMA has the same VH12 heavy chain gene as the VRC01 class antibodies, but it has a different light chain. So I'll come back to that on the next slide. So what we've done here is tested for binding to recombinant monoclonal antibodies that could be VRC01 precursors. What we wanted to know is, can we actually identify, now I'm frozen, true VRC01 precursors in naive PBMC samples. And so the way that we do this is we label our anti-idiotype with a fluorophore, and then we can stain B cell receptors on naive B cells from HIV naive donors. We then use FACS to single cell sort those, RT-PCR to sequence the antibody, or to amplify the antibody heavy and light chain genes, and then we sequence them and ask if those are VRC01 precursors. And so this will tell us if they can, in fact, recognize true VRC01 precursors in naive PBMCs. And it also tells us the rationale behind this is that the B cells that the idiotype stains are those that will presumably respond to vaccination. And so this is a summary of those results from our B cell sorting experiments. And so what I'm showing on the left graph is the percentage of B cells that we sorted with each individual idiotype. So we either sorted with IV 1, 2, 3, 4, 8, 9, 12, or 13. Each dot represents a different donor that was sorted. And then we sequenced, we looked at the frequency of B cells that had either the VH1/2 heavy chain gene or the 5 amino acid CDRL3. And then we also compared bulk sequence data that was unbiased. And so you can see the VH1/2 is normally present at a frequency of about 2%, and a 5 amino acid CDRL3s are quite rare. They're present at about less than 0.5% of the antibody light chain sequences in a naive repertoire. So what we found was we were able, with these two idiotypes, IV9 and IV12, to strongly enrich for antibodies that express the VH1/2 heavy chain. So roughly 90% of the B cells had it. But if we looked at the light chains that were associated with those two antibodies, none of them had the VRC01 class light chain. In contrast, this idiotype IV4 strongly selected for B cells that had a 5 amino acid CDRL3, but those B cells were not paired, or the B cell receptors were not paired with the VH1/2 heavy chain. And so we asked if we could combine the specificity of these two of these different idiotypes to get something that might more readily engage VRC01 class B cells. And so what we did is we made Fab versions of the IV9 antibody and the IV4 antibody, and then we fused those at the C terminus to either a spy tag or a spy catcher molecule. And these, what happens when you take these two proteins and mix them together is that they form a spontaneous, they spontaneously form a covalent bond in a 1-to-1 ratio. So these now become physically linked together. And you can combine the specificities of the two antibodies, and by purifying them both independently and then mixing them and get this nice well-defined molecule. And this is very similar to sort of a bispecific Fab2 molecule. And I should note that when we did this B cell sorting, we sorted a roughly 1,100 B cells and only found one VRC01 class precursor. So we then carried out sorting experiments with the IV4, IV9 bispecific molecule. And there we sorted 488 cells, and we're able to sort out only 3 VRC01 class precursors, which isn't exactly what we were hoping for, but it's still better than what we got when we sorted with any of the other idiotypes alone. But we're not entirely, I'm not entirely convinced this tells the full story, because the way that we designed our molecule to be bispecific has a potential advantage, and that is illustrated by this cartoon. So if you had, let's say, a naive B cell that had a germline VRC01 class light chain, but a non-VRC01 class heavy chain, it could be bound by the light chain specific arm of the anti-idiotypic molecule. Similarly, if you had a B cell that had a VH1/2 heavy chain, but a non-VRC01 class light chain, it could be bound by the heavy chain arm of the bispecific molecule. Whereas if you had a cell that had both, the heavy chain specific arm could bind the heavy chain, and the light chain specific arm could bind the light chain on an adjacent B cell receptor. And then this would allow those B cell receptors to cross-link and trigger B cell activation. So these experiments, although what we would be able to do is sort all of these particular B cells because we're using a fluorescent bait that could bind the cells, but we think that only those that are true VRCO1 class precursors will be readily activated using this approach. And so we wanted to test that a little bit more directly. And so David Namazi at Scripps had developed some germline VRCO1 knock-in mice. And so what he did is he knocked in the VRCO1 light chain into the murine kappa locus. So all of the, in a homozygous mouse model. So all the B cells in the mouse express the germline VRCO1 light chain, and they compare with random murine heavy chains. And then he also had another mouse line where he knocked the VRCO1 heavy chain, the germline VRCO1 heavy chain into the heavy chain locus, and then those compare with random mouse light chains. And then we crossed those two mice to make a mouse that was homozygous for both. And what we did is we took our fluorescently labeled anti-idiotic antibody and then stained those cells. And you can see that all or most of the cells stain positive with the anti-idiotype molecule. So this reflects what we saw in our sorting data, that it can definitely stain non-VRCO1 class B cell receptors. But when we look at B cell activation through calcium flux in these different cell lines, despite being able to bind, the bispecific molecule only really activates the cells that have both the heavy and the light chain. So suggesting that the way we designed this could potentially have a selective advantage to activate the B cells we want. So we then moved on to testing this in vivo. And what we did here is we took the knocked-in mice that have both the germline VRCO1 heavy chain and light chain, and then took B cells from that mouse, labeled them with a cell tracking dye, cell trace violet, and then transferred them into CD45.1 wild type mouse. And we do this at the approximate frequency that we know that VRCO1 class antibodies are present at in humans. And by virtue of congenic marking, we can tell whether the cells came from the donor mouse or the recipient mouse. And so what we did was then we just sort of repeated our in vitro experiment, and we compared immunizing with either the heavy chain specific Fab, the light chain specific Fab, or the bispecific. And so what I'm showing here is after 5 days after immunization, we're looking at specifically CD45.2 cells and looking at whether or not those cells divided in response to immunization. And so you can see animals immunized with PBS, there was basically no cell division. We see a little bit when we immunize with either monomeric form of the heavy chain or light chain specific idiotype, but almost all the cells divide when we immunize with the bispecific. And that's just quantified across several mice here. And then we also look 14 days after immunization at the frequency of the CD45.2 cells or the knocked-in B cells in both the spleen and lymph node, and we see roughly a 10-fold expansion of those relative to mice that were just immunized with PBS alone. So this suggests that the bispecific can more readily activate the cells of interest in vivo, and then it can induce them to expand up to 10-fold after 2 weeks. We also wanted to look at how well these specifically target the B cells we want. And so we do an assay where we looked at, first we took the mice and we gated on germinal center B cells by virtue of BCL6, GL7 gating. And then within that population, we stained with our fluorescently labeled anti-idiotype to identify all the cells that are antigen specific in that GC. And then within that population, we asked whether they were CD45.2 or VRCO1, or if there were other off-target B cells coming from the endogenous mouse B cells. And so I'm just sort of showing a summary of that data here at the bottom, where we look in the spleen and the lymph nodes. At the germinal center, the frequency of antigen-specific germinal center B cells in animals immunized either with the bispecific or just a PBS control. And you can see that 10 to 15% of the antigen-specific germinal center germinal center cells are antigen-specific in this case, and that the majority of them are of the CD45.2 rather than the CD45.1 variety. So suggesting that these responses in the GC are primarily on target. And so to summarize, we've generated these anti-idiotypic monoclonal antibodies against VRC1 class precursors. Unfortunately, we didn't find any that were broadly reactive with several VRC1 precursors, but we did show that we could develop a bispecific platform and leverage it to selectively target and activate B cells expressing in particular VHVL pair. And that this can actually activate our target cells with minimal off-target immunogenicity in vivo. And so now what we're working towards is sort of coming back and testing the hypothesis that we had earlier, that we could, prime with the anti-idiotype and then boost with an envelope, and that's going to have some kind of advantage to just immunizing with envelope alone. And so we're pursuing this in both the adoptive transfer model that I talked about already, and we also have access to a fully humanized mouse, or a mouse that has the majority of the human antibody genes knocked into the mouse locus. So this almost acts as a surrogate for immunizing humans. And in addition to that, we're developing anti-idiotypic antibodies against other neutralizing antibody classes that have well-defined genetic signatures, sort of like VRC01 does, targeting both other epitopes on HIV-1 as well as antibody classes against other viruses as well. And so that's sort of the first part of my talk. And I'll maybe stop now and take any questions anybody has before I move on to the second bit, because it's a little bit different.
Andy, I have a quick question, and anyone else feel free to put it in the chat and I can read them, or you can turn your video on and jump in like I'm doing now for a question. My question, I don't know if it's like a naive one or not, but one thing I wonder about with the anti-idiotype priming is how are you engineering T cell help in this setting? And if you guys like thought about conjugating antigen to the bispecific or something to try to drive that, or have you?
Yeah. So just in the interest of time, I really didn't discuss that, but it's an important thing, and we definitely do that. So we've actually we've shown this now in a few different mouse models, and I only really spoke about the one today. But what we found is if you take a full-length idiotypic antibody and immunize with it to target your B cell, what happens is they'll initially expand, but then they die off. And so what we aren't entirely sure of is whether that's an FC-mediated thing, so you're recruiting some other effector cell to kill it, or if there's just insufficient T cell help on the molecule. So what we did is we made this, in this case, we made a Fab2 version, but we've also made like multimeric Fabs of anti-idiotypic antibodies that are fused to another protein. And often what we'll do is we'll put a peptide on it that we know elicits a strong CD4 response in mice. And then that will basically rescue and cause the cells to like proliferate and divide. So we do engineer a T cell epitope in if there isn't sufficient T cell help. But in some cases, like if the spy catcher spy tag also is going to provide, it's also foreign antigen, so that's going to provide some T cell help as well. But it's something that is very important for sure.
Yeah, I mean, another question, I guess, related to that is if you had like an envelope peptide, would that promote a better secondary immune response or something than a non-specific, just due to T cell memory that might contribute?
Totally. So we've thought about that for sure. And what we've done instead, at least in the pilot experiments we've run with these sort of prime boost examples that I'm talking about here, is we would put a common peptide on both of them, the idiotype and the envelope, so that there is some T cell memory. But yeah, I think, we could, there are a lot of examples of sort of universal or fairly conserved envelope T cell epitopes we could put on there as well.
Super cool.
I had a question. I was wondering, is there much known about the diversity of IgG subclasses in the human bNAbs? And is that something that you guys are you trying to target a particular IgG constant region? Or is that being considered at all?
No, I mean, so I guess to answer your question in two parts, some bNAbs are associated with particular isotypes. So the antibodies that bind to the base of the spike, or the anti-MPERs, tend to be IgG3, whereas a lot of the other ones are mostly IgG1. So, well, but what we aren't targeting constant regions. I mean, what we're targeting is the variable regions, but we're targeting them non-naive cells. So those would be obviously IgM. And then the idea would be, I mean, ideally we could get a polyclonal B cell response to come out of those that has multiple isotypes. But we're not specifically targeting one over the other. We're targeting the variable regions.
Thanks.
Yeah, you're welcome.
Andy, have you tried to see whether this strategy that you did in the knock-in model would generate a memory B cell population following your
Yes.
terminal center response?
Yeah. So again, not in this study, but in a previous one we did that I didn't talk about. We did phenotype the B cells, and they were primarily plasmablasts, but there were memory B cells that were elicited as well.
Because is the strategy designed to generate, like, expand memory, and then that would be the precursor that you want to target? Is that
Yes.
That is the strategy, right?
Yes, I believe so. That's what we would want to do. So I mean, if we could basically like increase the frequency of memory cells that come out of it, then those would be the ones we'd presumably be able to boost.
And why do you think you got proliferation with either the light or the heavy chain only knock-in?
They're probably multimerized on FDCs, is my guess. Right, so you'd have the antigen to be presented on FDCs, and then it could engage multiple BCRs.
Right.
Yeah.
But wouldn't that be an issue in general?
Yeah, it could be. I mean, ideally it's kind of the best molecule we have, but it's not perfect.
Thanks.
Andy, it's Lisa Frankel.
Hey, Lisa.
Hi, very nice talk. I'm wondering, did you look at all to see if T cells were stimulated, and specifically if there's any responses, and if so, what type?
We did not.
Do you think it would, given the spike antigens and things, do you think that these could induce T cell responses?
I mean, it would, but I don't think that they'd cross-react with envelope in any way, right? Because they're just so distinct.
Okay, thanks.
Yeah, you're welcome. Okay, so I will move on then and talk about the other thing that we've been working on, and that is vaccine development for Epstein-Barr virus. And so, of course, this is a ubiquitous virus that infects greater than 90% of the adult population. And it often causes infectious mononucleosis when you first get it, which is due to an expansion of infected B cells and an expanded T cell response that's trying to kill those off. And that usually self-resolves, and most people carry it asymptomatically for life. But EBV is associated with a number of cancers of both B cell and epithelial cell origin. And it's estimated that it's responsible for roughly 200,000 new cancer cases and 140,000 deaths per year. In addition to this, it's also a causative agent of lymphoproliferative disease in immunocompromised individuals, which is the result of reactivated EBV in the context of immune suppression. It can develop into a lymphoma that has a fairly high mortality rate. So it's thought that an EBV vaccine that could either prevent or reduce the global burden of malignancies and morbidity associated with EBV infection would have a significant global health impact. And so the virus itself has tropism for both B cells and epithelial cells, and it's orally transmitted. So the virus comes in through saliva, and then in the oropharynx, both cells that are susceptible, epithelial cells and B cells, are present. So potentially the virus could come in and infect both cells. So we think an effective vaccine, or at least a prophylactic vaccine, would need to inhibit or severely limit replication or infection of these two cell types in the oral cavity. And when it infects these two cell types, what's interesting about EBV and herpes viruses in general is that the attachment entry proteins functions of attachment entry are spread across several virally encoded glycoproteins. And so this is an example of, or this is how EBV enters epithelial cells. So the first step is an attachment of the virion to integrins on the surface of epithelial cells, and that's through that interaction is mediated by a viral protein called BMRF2 that binds to integrins. Following attachment, two glycoproteins encoded by the virus called GH and GL bind to a cellular receptor. It's thought that it's FA2, but there's potentially other receptors as well. And this binding to the cellular receptor causes the GHGL to undergo a conformational change that allows it to trigger the fusion protein, which is called GB. And once GB becomes activated, it can refold to embed its fusion loops in the host cell membrane, and then refold to promote the merger of the host and viral membranes. And this is like very different from HIV, where there's only one protein involved. So in this example, there's at least four proteins involved in the entry pathway. And for B cell infection, the proteins that are involved are also different. And so the initial attachment step is mediated by the viral protein GP350, and that binds to either CD21 or CD35, which are complement receptors 1 and 2. And then after attachment, the GHGL complex is again involved, but it's associated with another viral protein called GP42. GP42 then interacts with MHC class 2 on the B cell surface, and then it is thought to cause GHGL to undergo a similar conformational change to what happens during epithelial cell infection that leads to the triggering of GB. And so despite all of these proteins are involved in infection, but so far subunit efforts for EBV vaccines have focused on only one of those, and that is the GP350 protein, which again mediates attachment of the virus to either complement receptor 1 or 2. And so the most advanced trial of a GP350 vaccine or any EBV vaccine was a phase 2 trial in adolescents where they were given 3 doses of recombinant GP350. And these were all EBV negative adolescents. I believe they were in the Netherlands. And what they observed was a significant reduction in the incidence of people who experienced symptomatic infectious mono in the vaccine arm relative to placebo, but overall there was no difference in the infection rates. So these data suggested that there may have been a clinical benefit to having an EBV vaccine, but they also suggested that the vaccine efficacy might be improved if other antigens were included. And so I mentioned there's quite a few other antigens involved. And so we were interested in looking at which of these might be good targets for antibodies and which might be ideal vaccine candidates. And we sort of, after working in HIV at the time for about 6 or 7 years, we wanted to apply some approaches that had sort of really revolutionized the field at the time, and that was the identification of a lot of these monoclonal neutralizing antibodies. And basically the idea is that if you have a EBV infected donor or a donor infected with anything, you can isolate antigen specific B cells through flow cytometry or root B cell culture, any method really. And then you can culture those, for example, screen for antibodies that have some kind of function that you want. And that could be just as simple as antigen binding or a neutralization assay or ADCC, for example. And then once you identify a B cell clone that's giving you the phenotype you want, you can sequence the antibody genes and make a monoclonal antibody. And then you can use that to define, for example, the protective epitope that could lead to epitope-focused vaccine design, or you can do all kinds of other stuff like use those antibodies for passive transfer studies, etc. And so when we started this back in 2015 was when we first started this work to just contrast what was known at the time for HIV. There was one target of neutralizing antibodies, and that's the envelope protein, whereas with EBV, there's at least 5 targets of neutralizing antibodies, or at least proteins involved in entry. For HIV, there's literally been hundreds of broadly neutralizing antibodies that have been identified from different infected donors. And we knew a lot about how these BNAbs interact with envelope. Whereas with EBV, at the time, there were only 5 neutralizing antibodies that have been described in the literature, and all of those have been isolated from murine hybridomas. And also there were no structures of neutralizing antibodies bound to any of the EBV glycoproteins. So we applied this approach to EBV, and we tried to get antibodies against all the viral glycoproteins that I mentioned earlier, but we particularly focused on GH and GL and GB because these are the 2 proteins that, well, GHL is 2 proteins, so these are the 3 proteins that are common to both the B cell and epithelial cell entry pathways. And so we were able to successfully isolate one antibody against GHGL, and that's AMO1. And so I'm just showing BLI traces here. And so when you see a signal go up on the y-axis over time, that's the antibody binding to the antigen. And so AMO1 shows specificity for GHGL, but none of the other recombinant proteins. And then we also isolated 4 other antibodies, AMO2, 3, 4, and 5, against the GB protein. And all of these were isolated from a total of 800 million PBMCs that was taken from 4 different donors. So these memory B cells that we were able to isolate were pretty rare in the memory B cell population. We had to do quite a bit of trickery to actually to bind and validate these, or to identify and sort these B cells, but I'm just going to skip over that for now, but I'm happy to talk about it if anybody's interested. And so we took these antibodies that we had and we put them into an in vitro neutralization assay of either epithelial cell infection or B cell infection. And you can see that in the epithelial cell infection assay, both AMO1 and AMO5 neutralized the virus infecting epithelial cells, whereas AMOs 2, 3, and 4 were non-neutralizing. And then if we put them in the B cell neutralization assay, the activity of AMO5 drops off significantly, but AMO1 still potently neutralized B cell infection. And so these were not only the first neutralizing antibodies described against EBV that have been isolated from humans. AMO1 was the first antibody that has been described that could potently neutralize infection of both cell types. So obviously it's an interesting antibody, and we were interested in knowing where exactly it binds on GHGL. So we worked with Marie Pansera at Fred Hutch. And so she solved the crystal structure of the AMO1 Fab. And then David Wiesler's group at UW solved the cryo-EM structure of AMO1 bound to the GHGL GP42 complex. And we were able to fit Marie's structural data into that to get a high resolution map of the epitope. And so we know it bridges across 2 of the domains of GHGL and interacts with both GH and GL. So we have a really good idea of where the epitope is and could potentially use this in the future for epitope-focused vaccine design. And I'll mention that some of you may know Michael. I think he spent some time over at Children's, but he was the person in my lab who actually isolated these 2 antibodies. And so one of the key questions that we were interested in, and now that we had these cool neutralizing antibodies, was trying to address the question. It's kind of an open one, can antibodies even protect against EBV acquisition? So I've kind of been speaking about this in a way, because I'm a neutralizing antibody person and interested in neutralizing antibodies. But the question is, can these things even protect against EBV acquisition? And if so, then that would provide a good rationale that these are a good target for vaccines. And so there's evidence in the literature for both. And some of the most compelling evidence comes from studies of infants in Sub-Saharan Africa. And so these infants are usually EBV positive by the time they're 1 year old, which is very different than what we see in North America, where most people get it during adolescence. But all of the kids are born EBV seropositive because they get antibodies from their mother. And then those wane over time until they're about 6 to 8 months old. And then the kids will test seronegative for a few months and then eventually seroconvert. And so it suggests that the maternal antibodies had protected them from EBV infection over time. And this, in more recent studies where people look at the virus, viral DNA rather than serum antibodies, they rarely ever detect EBV infection in infants prior to 6 months of age. So that strongly suggests that antibodies are protective, at least in these infant populations. But there are, on the other hand, several studies where people have identified what they call different EBV strains in infected individuals. And some people have suggested this could mean that even if somebody had been previously infected and developed antibody responses or T-cell responses, for example, those still were unable to protect them from infection with an additional strain or prevent superinfection, in other words. Of course, there's several caveats to both of these because they're observational studies, and so we wanted to address this a little bit more experimentally. And so the first thing we did was we worked with David Rawlings over at Children's there, and Swati in particular in his lab. And we knew that David worked quite a bit with these NSG mice in which you can engraft human lymphoid progenitors. And so when you do this, the mice will develop human lymphocytes, the B cells of which can be infected with EBV. So we would do the engraftment and then wait for the cells to engraft for 8 to 12 weeks. Then we would inject our neutralizing antibody or controls, for example, and then wait 2 days and then challenge them with virus. And then we monitor the mice for a period of 8 to 9 weeks. And by taking blood draws, monitoring weight, etc. And so these are some of the results of that study. And so the top, I'm showing the viral load in blood. So we sample blood from the mice every week for 9 weeks. And then we had a group of animals that remained uninfected, so we never challenged them with virus. And then we had another group that we challenged with virus, but never gave any antibody to. And you can see they all become viremic by about 4 or 5 weeks after challenge. Whereas the uninfected animals remain below the limit of detection or quantification. And we had another group that we gave an isotype control, so we gave them VRC01, so that's the anti-HIV antibody that doesn't do anything against CBV, or we gave them AMO1. And so we gave 18 mice AMO1 and all challenged them with the same amount of virus. And you can see that all of the animals that received the control antibody became infected, but only 2 of the animals that had been previously given AMO1 showed detectable viremia in the blood. We also, at the end of the study, euthanized all the animals and looked at viral load in the spleen. So here we can sample quite a few more B cells than we can sample in the blood and get a little bit more sensitivity. And you can see that the 2 animals that became viremic are shown in triangles. So those clearly became infected. And relative to the controls, we saw slightly higher levels of EBV in the AMO1 treated group, suggesting that it looks like the EBV was probably largely blunted or severely limited in its ability to infect cells, but there may have been some low-level breakthrough infection in these animals, especially if you compare that to the clearly infected control animals. And so this was very encouraging. So it just suggests that there's a pretty strong antiviral effect by having the presence of the antibody. May or may not have achieved sterilizing immunity, but one thing to keep in mind is that this animal model is not perfect because the murine B cells can't be infected with, or the murine epithelial cells can't be infected with EBV. And so the virus, as a result of that, the virus has to be given IV to establish infection, which is not the normal route of transmission in humans. So in parallel, we were working with the EBV ortholog called rhesus lymphocytovirus. And so this is the ortholog that infects macaques. So every EBV gene has an orthologous gene in the LCV genome. EBV can infect macaques and LCV can infect humans, but they are the sort of equivalent viruses for those two species. The virus is orally transmitted in monkeys the same way it is in humans, and the course of infection is very similar. And for us, importantly, the GH and GL proteins were highly identical. They're greater than 85% identical at the amino acid level. So we did some quick experiments to ask if AMO1 could cross-neutralize rhesus LCB. And so here I'm just showing fluorescently labeled AMO1 staining either untransfected 293 cells or 293 cells transfected with rhesus LCB GHGL. So we do see positive staining showing that they cross-react. And then we sort of did a bit of a crude in vitro neutralization assay where we took macaque B cells and then we gave them, challenged them with rhesus LCV in vitro, and then we stained for the presence of EBNA2, which is one of the first viral proteins that's made after infection. And so roughly 7% of the cells stained positive for the LCV in this assay. If we gave AMO1 prior to that, none of the B cells became infected. And then if we gave AMO2, which is the non-neutralizing control, roughly the same number of cells were infected in LCV versus the AMO2 group. So these suggested that AMO1 could cross-neutralize and cross-react with LCV. So we were able to fortunately acquire some money from the Primate Center through a pilot study to do a challenge study. And so what we did is we got 6 infant macaques acquired shortly after birth. They were hand-reared in a colony, or away from the colony in a nursery in the Primate Center so that they never were exposed to LCV. We serially tested them over that time to make sure they were LCV negative and waited until the maternal antibodies waned. And then we gave the monkeys a dose of 20 milligrams per kilogram of either VRC01, the isotype control, or AMO1. And then 2 days later, we did an oral challenge with 50 transforming units of the virus. And the results of that study are shown below. And so we looked both in blood and saliva. So we looked for blood, B-cell associated virus and also viruses being shed in saliva. And then we also did looked for seroconversion to the viral capsid antigen. And so you can see after challenge indicated by the black arrow, both of the animals in the control group became infected. We could detect virus in both blood and saliva, and those 2 animals also seroconverted over the course of the study. But one of the animals that received the AMO1 antibody, this animal A18155, he became infected. We detected virus in both the blood and saliva. And so we then asked if there was something different about this animal, maybe why was he infected and the other ones weren't. And so when we looked at the actual levels of the antibody in the plasma at the time of challenge, so this is using an antigen-specific ELISA, where we're looking at the binding of AMO1 to GHGL. You can see that the 3 animals that were protected had much higher levels than the animal that became infected, and those decayed slowly over time. They were present for roughly 3 weeks. Whereas the animal that was infected had really low levels, and they waned to undetectable levels after a week. Similarly, at the time of challenge, the neutralizing titers in the serum of the animal that was infected were about a log lower than they were in animals that were protected. So collectively, these data sort of suggest that neutralizing antibodies can protect against oral transmission of lymphocryptovirus in the rhesus model, and it also demonstrates that we can use the mouse model as sort of a more high throughput challenge model to evaluate antibody protected immunity in humanized mice against EBV. And so what we're working at now is trying to ask if we can elicit these types of antibodies through vaccination. So I'll just quickly go through what we've been doing in the last little while. And so what we've been doing is trying to develop these self-assembling GHGL-based nanoparticle vaccines And so we do that by basically fusing a C-terminus of the GH ectodomain to multimerization domain and then co-transfecting with GL and then purifying what comes out. And so we've developed GHGL that forms a tetrameric nanoparticle, a heptameric nanoparticle, 24-meric nanoparticle, or a 60-meric nanoparticle. And these are several collaborations with various groups, several in Fred Hutchinson, as well as at UW and Neil King's lab, who helped us develop this 60-mer. And so this is just showing the immunogenicity of these nanoparticles in mice. So we took regular black 6 mice, immunized them 3 times, and then just bled them 2 weeks after immunization and looked at their endpoint binding titers to GHGL. So you can see after 1 shot, all the nanoparticles do better than the monomeric GHGL by quite a bit. The 60-mer was the clear winner in this experiment after 1 shot, and then after the second immunization, all the titers were boosted. Again, the titers elicited by the monomeric GHGL were lower, and then after we gave 3 immunizations, they kind of all leveled out. But there appeared to be a clear advantage to immunizing with the nanoparticle at least 1 or 2 times, and that the 60-mer consistently gave the highest binding titers. When we look at neutralizing titers, we see a similar story. So here's just looking at the ability of the mouse serum to neutralize EBV infection of epithelial cells. You can see that the 60-mer does the best after 1 immunization. We don't get any neutralizing titers after a single immunization with the monomeric GHGL. After 2 immunizations, everything's boosted again, and then they sort of even out after the 3rd. And a similar thing is seen when we look at B-cell neutralizing titers, except the difference is that it took at least 2 immunizations to get any neutralizing activity against B-cell infection. And again, this sort of highest valency 60-mer nanoparticle does better than all of the other ones, at least after 2 immunizations. So what we then did was we did set up a challenge experiment. What we did is we immunized regular black 6 mice 2 times with either the 60-mer or the monomeric GHGL. And then we purified IgG from those animals and then gave equal amounts of that purified IgG to humanized mice. And so this is necessary because, of course, the mice are immunocompromised and they don't really support B-cell responses or good B-cell responses in response to vaccination. But it's also advantageous for us because we can say that any protection we see in the mice is coming from antibodies. So then after transfer, the mice are challenged. And what I'm showing here at the bottom is just the titers of GHGL antibodies after transfer. And so we had 3 groups. One group was immunized with had IgG from mice immunized with the 60-mer. Another one received IgG from mice immunized with the monomer. And then we had a control group that just got IgG purified from a naive mouse that hadn't been immunized at all. And you can see we get much higher binding titers to GHGL on animals that received IgG from animals that were previously immunized with the nanoparticle. And then what we did is we just looked in this study at survival. So we let this one go to 10 weeks. And there's 5 animals per group here, except for the monomer group, which only had 4 animals in it. So you can see in the infected control group, all of the mice eventually died after about 56 days post-challenge. Similarly, none of the mice in the naive group, so these are mice that received unimmunized IgG from unimmunized animals, they all perished by the end of the study. 1 of 4 animals survived that received the GHGL monomer, but all the mice that received the 60-mer survived, as did all the animals in the uninfected control group. So these are pretty encouraging results, and we're just doing a few follow-up studies here and hoping to publish this pretty soon. But to summarize this part, we've isolated the first EBV neutralizing antibodies from humans. We identified the AMO1 antibody and its epitope as a critical site of vulnerability on EBV GHGL. Showed that passive transfer of AMO1 can protect against EBV challenge in mice and non-human primates. That the GHGL nanoparticle vaccines elicit superior immunogenicity to the monomeric GHGL, and that the multivalent display may be necessary for protection, at least in the high-dose humanized mouse challenge model. And so what we're working on now is testing the GHGL nanoparticle vaccine in a rhesus model. And then we're also starting to look at how mRNA delivery affects the antibody responses to GHGL, both as a monomer and as a nanoparticle. And so I'll conclude there. I have a lot of people to thank from my lab. There's probably too many people to mention. But Emily, Andy did a lot of the work with the the idiotype stuff in mice. Harmon's been driving a lot of the EBV work, particularly the vaccine development. We worked with Marie's group a lot for structural studies. We started the idiotype work with Justin and Leo's groups as well. We rely pretty heavily on the antibody technology core at the Hutch. Great collaborations outside of the Hutch, including with David's lab in particular. We worked really closely with Swati on developing a humanized mouse model for EBV infection, and then external collaborators at Scripps as well. And of course, our funders, who we couldn't do anything without their support. So thank you, and I apologize for going over time, but I appreciate everybody for hanging around.
Thank you, Andy. That was an awesome talk. If anyone has any questions, please send them to the chat. I can start off with one you might have heard before. Why do you think the high valency immunization is like promoting such a bigger response, in the prime and the boost setting?
I think, I mean, there's several reasons. For glycoproteins, it's been shown that the multimeric display helps. They get to the lymph nodes better. They just get traffic to the lymph nodes better. You also get multiple engagement of multiple BCRs at the same time, so you get strong B cell stimulation. With the nanoparticle, if you think about it on a molar level, if a B cell internalizes one copy of a GHGL monomer, it's got one copy of all the T cell epitopes it could present. Whereas if you have a 60mer, it can internalize 60 copies and present all 60 of those to T cells as well. So that's part of the reason. But honestly, it's kind of a black box. But it clearly seems to do much, much better than the monomers for several antigens that we've looked at.
Yeah, it's super interesting. It'd be cool to be able to deconvolute it. For sure.
Yeah, I mean, people are definitely working on it, and there's sort of all these different aspects of it that seem to be beneficial.
Just wanted to say congratulations on the 60mer data. That's awesome.
Yeah, it's pretty cool, right? We're really excited about it.
That is super cool. I mean, I don't know if people recognize how impressive that is, but that's really super cool.
Yeah, we're trying to get that out the door as soon as we can.
My son would have liked that before he went to college.
Oh, I think there was a question in the chat.
Yeah, I can read it.
Oh yeah, thanks. I can't see it anymore.
So this is from Ting Ting Zhang. So she wants to know, do you think the EBV vaccine strategy will also work for people that have latent EBV to protect against reactivation?
I think that's a really great question. And I think it depends on how, so in the context of immune suppression, for example, you can have either a monoclonal expansion of a latently infected cell that never really expresses the lytic antigens, in which case I don't think that the antibodies will do anything. If you have B cells that become lytically infected and start spewing out virus that are infecting new B cells that cause a disease phenotype, then the antibody might. So it's kind of I think my understanding is that most of the lymphomas you see are monoclonal, so I don't think that it would. But I mean, it's possible.
Do you think that you could engineer ADCP function in targeting some of the surface
Again, it depends on whether or not they even express the antigen, right? So
Yeah.
if it's there for sure, I think you could get potentially ADCP or ADCC or anything like that to work. But that's and I don't know if there's a universal answer for that because I don't think all the lymphomas are necessarily the same. But I mean, we've explored a little bit, and we're still looking at like whether we could give like ammo-1, for example, to knock down lymphoproliferation. And then I know some people at Oregon who have like a macaque model where they can see PTLD like expansion sometimes when they suppress monkeys for gene therapy studies, basically. And so we're thinking about trying to treat some of them just to see what happens.
could murinize some of the antibodies. See if effector function would
Yeah, we could.
mitigate the human lymphoproliferative stuff.
Well, yeah, I mean, we've thought about using the mouse model for that, but there's also like the strain that we use here is called B95.8, and it expands almost exclusively like a non-lytic replication, but there are other variants of the virus that replicate more lytically. So we could like think about testing both different strains and seeing what the outcome is. But yeah, it's a great question. And I don't really know the answer.
There is another chat question from the chat. This from Cade Ito. He says, "Thank you for the great talk. Have you looked at different antibody isotypes produced after immunization with the multimers? And is there like a skewing in some way?"
We didn't. I mean, we certainly could. It's just something we haven't really looked at. We're kind of more interested in the functional responses, but I think that would be a good thing to look at for sure. And I honestly don't know what we would see. I guess what I will say, I think, is at least with when we did the passive transfer study, we did protein G purification. So all of those antibodies that went into the recipient mice would have been IgG.
That's cool. I have like another question for the first part, and sorry to like belabor things, and it's like kind of just a mind noodling thing. Do you think, like, are you guys interested or looking, or is anybody that you know of looking into ways to elicit anti-cancer responses kind of in the same way that you're thinking of for the antiviral ones by idiotype activation?
We aren't, but I've heard, I think you can do it. Like, I think you can make an anti-idiotype against like a lymphoma, a BCR and a lymphoma and kill it, basically.
Yeah, I was thinking more like if you could, like use the panning approaches to kind of look at, like for melanoma, you could look at melanin or something like that and like identify a VDJ, germline
Oh,
VDJ
sure.
that
Yeah, yeah.
responds to that, and then vaccinate to make an immune response, like something like that.
We have not, and but I mean that would be cool, and I think that whole approach sort of like to be broadly applicable hinges on identifying these antibodies that have genetic features. So like I think with HIV, people have dug deep enough that they find them, right? And it came up pretty quickly in SARS-CoV-2. Like when people started looking at the responses to CoV-2, there was like a VH353 heavy chain antibodies like bounce with super high affinity to the RBD with almost like no mutation at all. So I think it may be possible that there are like antibodies like that could recognize melanin. It might be, or like anything really, but
Yeah,
somebody
right.
has
Like,
to go find them, right?
like you could conjugate a bunch of different, melanoma specific things and try to identify one that, something that has a germline pairing. It's cool.
Yeah.
Yeah, I guess I was surprised you hadn't thought about that for COVID, right? With certain conserved heavy chains and heavy light chain pairing.
The problem with at least the conserved ones that we know about with COVID is those are the ones that the virus immediately escapes from. So
Right.
yeah.
I guess we don't know yet about other conserved ones, but
Yeah, I mean, I think it'll happen. I can't remember us speaking with earlier, but it's the number of mAb specificities that people have like figured out with COVID in the last couple of years is sort of mind blowing.
you could imagine if there was like some epitope that wasn't as escape penetrant or whatever, that would be a nice, because it's going to be hard to do that with an mRNA for the protein, probably.
Yeah, I think so.
That could be your COVID grant that you don't want to write.
Yeah.
And the nice thing there, it's a parallel approach. It's like not in
Yeah.
with the current approaches.
I feel like the problem with the COVID grants is just like a pile on, like everybody's trying to get them. It's like almost harder than,
Oh yeah.
It's
yeah.
terrible.
The idiot type would be a very different approach than almost anybody else would be taking, I think.
Yeah, it's true. I've definitely thought about it, but I'm not sure I want to go there just yet, but we'll
Yeah.
see.
Looks like you have enough going on. So
Feels like I have enough going
Yeah.
on.
thanks for a great talk, Andy. That was
Oh, thank you all for
Great.
listening and hanging out, and thanks again for the invite, Rich.
Yeah, it was great to see this cool stuff. And I'll follow up with you. Thanks.
Yeah, great. Thanks again, everyone.
Bye.
Thanks. Have a good afternoon.
You too.